Good afternoon, everyone. Thanks for attending the seminar. I'm going to be the presenter of the QAAFI Science seminar today. My name is Eugeni Roura, and I'm a professor for the Center for Nutrition and Food Sciences in QAAFI. I would like to start with the acknowledgement of country. The University of Queensland acknowledges the traditional owners and their custodianship of the lands on which we meet today. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognize their valuable contribution to Australia and the global society. The seminar today will go up to 1 p.m. It is anticipated that we will have probably around 10 to 15 minutes of questions. So please, I would like to encourage you to use the QA tab. Remember the QA tab and not the chat. We will be looking at the QA tab for your questions. And I encourage you to do that whether it's during the presentation or not, that's all right. But we will be attending the questions at the end of the seminar. So without further ado, I would like to introduce the speaker today, Dr. Maximiliano Müller. He got his bachelor's degree in Veterinary Science at the University of Chile, so he's Chilean of origin. That was in 2015, while in 2016 he was awarded a scholarship from the Chilean government to attend UQ in a PhD, joining a PhD program in my group. Max was successfully finalizing his PhD earlier this year, and actually recently he got the certification, so congratulations to Max. Just about end of May of 2021, so very recently. So I've been supervising him during these years, and he's done a remarkable, very good research work on amino acids and how amino acids can affect appetite, which is the topic of his presentation of today. Max, during these 4 years of his PhD program, has been awarded a number of recognitions, prizes, and remarkably as the best student oral presentation of the International Symposium of Digestive Physiology of Pigs. That was in 2018, which is a very prestigious reward. So congratulations to him. And his excellence as an oral presenter will be put at test today. Without further delay, I would like to just welcome Maximiliano, and let's hear his presentation on dietary amino acids, gut sensing, and feed intake regulation in pigs. Max, the floor is yours. Max, you might be mute. Are you here? Max, hello? Max, you're in mute? Can you hear us?
Hello?
Oh,
yep.
There you go. Okay.
Yeah, apologies for that. There's a small problem in there. Hopefully you can hear me now and you can see me now. So, well, thank you very much, Eugeni, for the introduction. I would also like to start my presentation just acknowledging the traditional owners and their custodianship of the land on which we meet today, and pay my respects to their ancestors and their descendants. So I would also like to mention that I'm quite grateful for being given the opportunity to present at the QAAFI seminar today. And as Eugeni mentioned, today I will be talking about dietary amino acids, gut sensing, and feed intake regulation in pigs. So here's the layout for today's presentation. So I will start my talk just providing some background on the topic of amino acid gut sensing and appetite modulation in pigs. And then I will move to talk about the gap of knowledge on this area and the aim of this study. And I will follow that with just talking about the experiments that we conducted to test our hypotheses on this subject. And I will then finally wrap up the presentation with a short summary, as well as some of the implications of this study, which may be of relevance for the pork industry. So without further ado, let's begin. So when it comes to the pig production, incentivizing feed intake in the post-weaning period is of utmost importance, not only for the performance of the animal, but also as well for the gut health and well-being. Therefore, one of the most common dietary strategies that farms follow is the inclusion of highly palatable and easily digestible protein sources, and that is generally coupled with a cap in the content of crude protein and the use of synthetic amino acids, particularly the most limiting essential, such as lysine, methionine, threonine, and tryptophan, in order to incentivize appetite in these animals. The problem is that even when these kind of practices are frequently followed, we tend to observe a certain degree of stagnation on growth and feed intake in pigs on the days following weaning. This has been attributed to a variety of multiple factors, such as, for example, overconsumption and diarrhea, just to name a few. But little attention has been placed on the satiating capabilities that some of the nutrients in the diet, particularly amino acids, can have in pigs. Now, you may be asking yourself, well, why particularly amino acids? Well, that is because amino acids and proteins are more satiating than other nutrients, such as, for example, carbohydrates and lipids, when compared on a calorie-by-calorie basis. But how exactly do amino acids modulate appetite in pigs and other animals? Well, Mellenkopf and colleagues in the 1950s were one of the first scientists to propose a possible mechanism. They suggested that as levels of amino acids increase in blood, this will be sensed by the central nervous system, leading to the reduce of hunger and therefore the termination of a meal or feed intake. However, nowadays we know that this process is not as simple, and there is a variety of other mechanisms involved behind the appetite regulatory effect of amino acids, particularly within the gastrointestinal tract. One of such mechanisms is or are enteroendocrine cells. These cells play a pivotal role on the sensing of nutrients, such as, for example, amino acids, and the release of signals involved in the mediation of the hunger and satiety cycle, also known as gut hormones, as I highlighted on the bottom right part. Now, in the pig, there is around 12 or more gut hormones that have been described up to date. However, when it comes to the gut hormones that have been closely linked to appetite modulation, there is 4 of them, which are ghrelin, an orexigenic hormone mainly produced and released in the stomach. And then we also have 3 anorexigenic hormones. The first one is cholecystokinin, or CCK, which is released mainly in the proximal small intestine. And we also have glucagon-like peptide 1, or GLP-1, and peptide tyrosine, or PYY, which are mainly released in the ileum and the colon. But how exactly do amino acids induce the release of these hormones? Well, that seems to be through the activation of amino acid sensors present in the transmembrane of these enteroendocrine cells. In pigs and rodents, there is a small group which have been related to gut hormone secretion, which are mainly G protein coupled receptors, such as the calcium sensing receptor, or CASR in the case of aromatic amino acids. We also have the mamitase receptor, or T1R1, T1R3 in the case of some acidic and aliphatic amino acids. And finally, we have GPRC6A, which has been linked with gut hormone secretion after stimulation with some basic amino acids, such as ornithine, for example. However, besides G protein coupled receptors, there is a variety of amino acid transporters, such as the ones you can observe here, which have been described to be present in enteroendocrine cells. However, the role on gut hormone secretion is not certain yet. Therefore, this is a subject that is or requires further investigation. Now, when it comes to the effect of amino acids on gut hormones, there is a small group which have been identified, at least in rodents, to be robust stimulants of a particular of two satiety hormones, such as CCK and GLP-1. And these ones are arginine, phenylalanine, tryptophan, glutamine, leucine, just to name a few amino acids, many of which have also been shown to play or have a similar effect in humans. However, when it comes to the pig, little is currently known on this topic, as only recently have new studies emerged investigating the effect of amino acids on the release of the so-called satiety hormones. Some of these more frequent studies seem to suggest that branched chain and aromatic amino acids may be or may have a strong anorexigenic effect in pigs. So overall, we can say that the appetite modulatory effects of individual amino acids in pigs is a subject that remains largely unknown. And why is it important to study? Well, that is because current formulation or dietary formulation practices seems to disregard potential excesses of non-essential amino acids and include safety margins for some of the most limiting essential amino acids, such as lysine and methionine. Therefore, it seems to be the case that pigs are generally exposed to an excess content of free amino acids in the feed, which could potentially be detrimental for their appetite. Considering this, the overall aim of this study was to characterize the anorexigenic properties and some of the underlying physiological mechanisms of amino acids in winner pigs, with the final objective of improving current formulation guidelines in winner pigs and potentially even at other productive stages. So here we have a diagram of the experiments that were conducted to evaluate the effects of amino acids on appetite modulation in young pigs. In red, we have some ex vivo experiments, and in blue, we have some in vivo studies. I will be describing each one of them as I go throughout the presentation, starting with the initial ex vivo experiments, which look mainly to evaluate the effects of amino acids in intestinal tissues. So the overall objective of this initial experiment was to, first of all, map CCK and GLP-1 secretion along the gastrointestinal tract of post-weaning pigs. And second, well, to screen the anorexigenic potential of the 20 proteinogenic amino acids by evaluating their impact on the secretion of CCK and GLP-1 under ex vivo conditions. Now, we decided to look at the effects of amino acids on these two particular hormones for a couple of reasons. In the case of CCK, because it's the most vastly described anorexigenic hormone. And in the case of GLP-1, because besides being described as a satiety peptide, it has also been shown to have robust incretin effects. That means it stimulates the release of insulin, another hormone involved in long-term regulation of appetite. So we hypothesized that CCK will be predominantly released from duodenum, while GLP-1 will be mainly secreted from the ileum of post-weaning pigs, which is something that has been described in adult or finishing pigs. In addition, we also hypothesized that essential amino acids, in particular branched chain, aromatic, and some of the most limiting, such as lysine and methionine, which have been shown to play a pivotal role on appetite modulation in pigs, will result in the highest anorexia through CCK and or GLP-1 release in the winners. So to test this hypothesis, we perform a primary intestinal culture. In short, we collected intestinal samples from euthanized animals. We cleaned these intestinal segments in the lab, and then we stripped them off of their muscle layer by using a couple of tweezers, as you can observe in the picture. And then through the use of a biopsy punch, we collected equally sized samples, which we then placed in a 24-well plate filled with KRB buffer. These plates were incubated for a period of 1 hour, after which the content of each well was replaced with a treatment solution containing one of the 20 proteinogenic amino acids at a concentration of 10 millimolar, which is a physiologically relevant concentration, or a control solution which was just pure KRB buffer. The plates were then incubated for an additional hour, after which the supernatant from each well was collected for the subsequent analysis of gut peptides. So as I mentioned before we had a look at the effect of amino acids on CCK and GLP-1, we first needed to determine the secretion pattern of these two gut hormones. And as you can observe here, secretion levels of CCK at the top right corner were higher in the duodenum when compared to the other segments of the small intestine. While in the case of GLP-1, the highest levels were observed in the ileum. Therefore, duodenum and ileum were selected as the ideal intestinal segments to evaluate the effects of amino acids on CCK and GLP-1 respectively. So this next slide shows the effect of essential amino acids on CCK secretion in pigs, particularly in the duodenum, when compared to a control solution, which in this case is shown as the gray box on the left side of the graph. And as you can observe, among essential amino acids, only phenylalanine was the one that showed a significant effect on CCK secretion. Well, in the case of non-essential amino acids, here we found both asparagine as glutamine as being significant stimulants of the peptide. Of course, we did the same for GLP-1 in the ileum, and as you can observe here, among essential amino acids, arginine, isoleucine, leucine, methionine, and tryptophan all significantly stimulate the release of GLP-1. As for non-essential amino acids, the only amino acid that stimulated the release of the peptide was glutamate. Now, these results are quite interesting because they're telling us that for the most part, amino acids stimulated the release of one or the other peptide, which could potentially be explained by the differential expression of amino acid sensors along the gastrointestinal tract of the pigs. I mean, some of these sensors mainly expressed in the proximal gap, while others seem to be more prominent in the ileum and the colon. In addition, the capacity of certain amino acids to stimulate only CCK may suggest that these particular amino acids may have a faster effect on satiety when compared to those that stimulate the release of GLP-1 in the ileum. Even more, those amino acids that stimulated the release of both peptides, such as, for example, leucine and tryptophan, it could potentially mean that these amino acids could have a long-term effect on satiety through the release of CCK first in the duodenum and then later through the release of GLP-1 in the ileum. In the case of lysine, and considering its relevance in pig nutrition, we decided to further investigate its effect on both CCK and GLP-1 secretion. And as you can observe on these graphs, we have the effect of lysine at different physiological concentrations, except for 30 millimolar, on CCK in jejunum and GLP-1 in ileum. So lysine at concentrations of 20 millimolar significantly induced the secretion of CCK, but the same did not happen for GLP-1 at 20 or even 30 millimolar. Now, we decided to investigate the effect of lysine in the proximal jejunum rather than in the duodenum for a couple of reasons. The first one is that when exposing jejunum and duodenum to lysine, we observe a high response from the proximal jejunum. And second, because the proximal jejunum is another intestinal segment with a high expression of CCK, only second to that of the duodenum. Now, these results suggest, therefore, that the mechanisms behind lysine-induced CCK secretion are primarily present in the jejunum rather than in the duodenum, which could potentially also be the case for other amino acids. However, this is a subject that merits further investigation, as we currently don't know if that is the case. So in conclusion, we can say that CCK and GLP-1 secretion is higher in the duodenum and ileum of weaned pigs, respectively. Second, well, that the most significant stimulants of CCK identified are phenylalanine, lysine, glutamine, and asparagine. And in the case of GLP-1, we found leucine, isoleucine, arginine, glutamate, tryptophan, and methionine as significant stimulant of this particular peptide. So overall, this study provided new information regarding the effect of amino acids on gut peptide in pigs, but had some limitations derived from the use of an ex vivo model. And that is because ex vivo model lacks many of the neural and hormonal signals present in live animals that could potentially enhance or inhibit the secretion of gut hormones and therefore the modulation of appetite. Considering this, the next step then was to corroborate the effect of some of these amino acids in live pigs. So we selected 5 amino acids, which were glutamate, isoleucine, leucine, lysine, and phenylalanine. And this was based on these results, as well as previous literature on gut hormone secretion, as well as the relevance on pig nutrition. So going back to this diagram of experiments one more time, Following the results of the initial ex vivo study, we then performed a couple of in vivo experiments to further evaluate the effects of some of the selected amino acids on appetite modulation in young pigs. So the objectives of these experiments were two. The first one was to characterize the impact of the selected anorexigenic amino acids on feeding behavior. And the second one to evaluate their impact on plasma CCK and GLP-1 levels in young pigs. So we hypothesized that oral administrations of amino acids identified to increase CCK and or GLP-1 secretion would result in higher satiation and satiety, measured as long-term feed intake, reduced meal duration, and increased intermeal interval in young pigs. In addition, we also hypothesized that increased plasma levels of CCK would be observed following the oral gavage of phenylalanine, lysine, and to a lower extent lysine, whereas in the case of GLP-1, concentrations will be enhanced with the administration of a branched chain and a glutamate in young pigs. So to test this hypothesis, we performed what is called an oral gavage procedure. Animals were fasted overnight, after which the animals were removed from their respective pens and put in a different room where they were lightly anesthetized with the use of isoflurane, which was administered via face mask, as you can observe on the picture. And then the oral gavage was given to the animals through the use of extension plastic tube of approximately 30 centimeters, which was attached to a 60 ml syringe. And following the gavage, animals were placed back in their respective pens for the subsequent evaluation of feeding behavior or the collection of blood samples, as you can observe on the picture on the right. So for the feeding behavior, particularly for the analysis of meal pattern, we placed cameras on top of each pen, which allows us to then determine if there were differences on feeding behavior among the different treatments. In the case of the blood sampling, we surgically catheterize animal in the external jugular vein, which allow us to collect samples at multiple time points, as shown here in this diagram. One sample was collected before the gavage, and then the other samples were collected at 5, 15, 30, 60, and 90 minutes post-treatment. If you can observe the gray square on top of the times, it shows all the treatments that were tested in the pigs. So we tested 5 different amino acids at concentrations of 3 millimolar per kilogram of body weight. And besides the amino acids that I previously mentioned, we also included glucose as a positive control for GLP-1. And we also had a negative control, which in this case was just pure water. So this next slide shows the results for feed intake in pigs following the oral gavage. And here I'm just showing the treatments that had a significant impact on this performance parameter. As you can observe, both lysine and lysine significantly reduced cumulative feed intake up to 3 hours post-treatment. While in the case of isoleucine, we observe a delayed effect on appetite as the amino acid only reduced feed intake between 2 and 2.5 hours post-gavage. Now, of course, we also had a look at the meal pattern of pigs following the procedure, and here I have a lysine and lysine. And here are the changes of meal pattern when compared to the control. So everything is shown as percentage of the control, being the control the dashed line at 100%. So each graph has 5 acronyms in the bottom. From left to right, those acronyms represent latency to first meal duration, intermeal interval, second meal duration, and finally a number of meals. So as you can observe, lysine significantly reduced the first meal duration and tended to increase the intermeal interval. Where in the case of lysine, we here observe an increase of the intermeal interval and a reduction of the number of meals. Now, the meal pattern changes as well as the feed intake changes observed for both lysine and lysine indicate that these amino acids are highly anorexigenic, and that potentially the mechanism behind their effect are probably similar, if not the same. And this potentially might be related to the release of gut hormones, and that is because gut hormones has been linked with meal pattern changes as well as a rapid reduction on feed intake, as was observed for lysine and lysine, but not so much for the case of isoleucine. As I mentioned before, this amino acid did not alter milk pattern and only had a delay effect on appetite, suggesting therefore that potentially the anorexigenic effect of isoleucine is unrelated to gut hormones in this experiment. Now, to properly corroborate if gut peptides were involved in the anorexigenic effect of lysine and leucine, as I mentioned before, we look at the blood kinetic of these gut peptides following an analog batch in pigs as well. And here we have the results for lysine and leucine. And as you can observe here, lysine tended to increase the levels of plasma CCK, as shown by the area under the curve. And in the case of leucine, we see a significant effect on plasma CCK when compared to the control solution. However, we did not see similar results for GLP-1 in the case of lysine and leucine, but we did observe that phenylalanine increased the levels of this peptide between 30 and 90 minutes post-gavage when compared to baseline values. Of course, we also had a look at our positive control, which here is shown on the right side, which is glucose. And as you can observe here, the glucose rapidly increased the levels of plasma GLP-1, but then these levels rapidly decrease again to baseline values within the first half an hour post-treatment, suggesting therefore that most of the glucose was potentially absorbed before even reaching distal segments of the gastrointestinal tract, where most of the enteroendocrine cells in charge of releasing GLP-1 are present. Now, the results for phenylalanine are particularly interesting because even though the amino acid increased levels of GLP-1, it did not alter feeding behavior or change meal pattern in any significant way. This could potentially mean that this amino acid stimulated the release of other gut hormones not studied in this experiment, such as, for example, ghrelin. Now, I'm mentioning ghrelin in particular because other aromatic amino acids, such as tryptophan, have been shown in pigs to be a robust stimulant of this appetite-inducing hormone. Potentially, therefore, the same could be the case for phenylalanine. However, of course, this is just speculation and is a subject that merits further investigation. So in conclusion, we can say that the oral gavage of lysine, leucine, or isoleucine reduced cumulative feed intake in young pigs. In addition, lysine altered meal pattern by reducing the first meal duration, followed by an increase in the intermeal interval. While in the case of leucine, this amino acid increased the intermeal interval and reduced the number of meals following the gavage procedure. Even more, leucine increased while lysine tended to increase CCK plasma levels. And finally, that glucose and phenylalanine increased GLP-1 describe two very distinctive time patterns, one of an early peak, while the other of a late increment, respectively. So taking all the results into account, lysine was shown to be the amino acid with the strongest effect on feed intake, and this anorexigenic effect was accompanied with changes in meal pattern and gut hormone secretion, which is something that we're expecting considering the relevant role of lysine on appetite modulation in pigs. So just as a summary of all the information that has been provided up to this point, we can say that as the pig feels hunger due to the activation of signals such as increased plasma levels of ghrelin and reduced plasma levels of glucose, feed intake is initiated. So as the animal consumes feed, amino acids are capable to interact with enteroendocrine cells present along the gastrointestinal tract, leading to the release of gut hormones such as CCK in the proximal small intestine and GLP-1 in the ileum and the colon. As shown by our results under ex vivo conditions, we found a handful of amino acids to be significant stimulants of CCK, such as phenylalanine, asparagine, glutamine, lysine, and leucine. And in the case of GLP-1, we found arginine, leucine, isoleucine, methionine, tryptophan, and glutamine. Now, in addition, some preliminary data from our group also suggests that a couple of amino acid receptors may be involved in lysine-induced CCK secretion, such as, for example, the metabolotopic glutamate receptor 1 and the cationic amino acid transporter 7A2. However, further investigation might be required in order to properly corroborate if this is the case. And when looking at the effects of amino acids in live pigs, we were able to corroborate the effect of 2 of these amino acids, which were lysine and leucine, on CCK secretion, but not so much on GLP-1. Now, CCK is capable of interacting with the central nervous system, particularly the hypothalamus, indirectly via stimulation of vagal afferents in the gastrointestinal tract, or directly through the bloodstream, leading to the sensations of satiation and satiety. Satiation generally refers to the termination of an ongoing meal, which is generally represented with a reduction of the meal duration, or in the case of satiety, this one generally represents a feeling of fullness, which is generally shown with an increment of the intermeal interval. Together, these changes in feeding behavior can lead to a reduce of feed intake, which, as I mentioned before, is not ideal in the post-weaning pig due to gut health problems and reduced growth performance. So having said that, I'm pretty much approaching the end of the presentation. Here I have a few implications that may be of relevance for the pork industry based on the results presented here. So the first one is that, well, dietary safety margins and levels of free amino acid should be reviewed and narrowed when possible, as to minimize any potential negative effect of excesses of lysine or other essential amino acids on feed intake and growth in pigs. In addition, current practices related to dietary safety margins of essential amino acids may lead to increased weight variation within a herd, that is, from all the way from post-weaning to slaughter, stimulating a higher degree of satiety in light versus heavy pigs due to differences in amino acid requirements. And finally, well, that our results are compatible with the relevant role of essential, but also as well non-essential amino acids on metabolism, having a potential impact on satiation and satiety, protein accretion and muscle growth, as well as limiting weight variation in the pig herd. And with that, I would like to, first and foremost, thank Professor Eugeni Roura, as well as Mike Rio, for their important contribution on a review paper we recently submitted on amino acid gut sensing and appetite modulation in pig in the Journal of Animal Production Science. I would also like to thank Professor Robert Van Barneveld, as well as Professor Alan Tilburg, for their contribution in the design of the experiment showcase here in this presentation. And besides that, I would also like to thank overall the Roura Research Group for their help, not only in the farm, but also as well in the lab throughout the different experiments. And last but not least, I would like to also thank APL for the overall funding of these studies. Having said that, I would also like to thank all of you for giving me the time of the day. So thank you very much for hearing me.
Great. Thank you so much, Max. Well done indeed. Good seminar. Fantastic. Now, we have received a few questions. Out of the ones saying, there's 2 questions which are actually pointing into the same direction, and they are inquiring about the relevance, the physiological relevance of the gavage that you use in terms of the concentrations, amounts. The levels that you have tested are biologically relevant. Are they important and can be inferred, the results could be inferred for the feed industry and the pork industry? Are they commercially relevant?
So yeah, good question. So the doses we tested in vivo were slightly above what could expect under normal commercial conditions. So, but the objective of this experiment was mainly to be a proof of concept, to showcase that amino acids have an effect on appetite, and that effect is guided by the release of gut peptides, and there's some implications on the meal pattern as it was showcased in the in vivo studies, related therefore the release of gut peptides with these feeding behavior changes in the pig.
Thank you. Well, obviously, you are using free amino acids when you do the gavage, and most of, not many amino acids will be present as free amino acids in pig diets. Can you maybe comment on that?
Yeah, that's a good question. Yeah, so we needed to test each amino acid individually in order to properly assess what was the impact of each amino acid. But yeah, it's true what you mentioned. There might be potentially some interactions under normal conditions that may occur when the animals consume the feed. So the animals, of course, the gastrointestinal tract come in contact with not only one amino acid, but a variety of these nutrients. Many of which may compete for the similar receptors, and therefore one may be negating the impact of others when it comes to gut hormone secretion. That could potentially happen, as well as potentially there might be some interaction that could lead to higher secretion of certain gut peptides. If you put, if the animal consumes high concentrations of, for instance, lysine, which was described to be an amino acid that stimulates CCK, but also lysine, this could potentially both together enhance the satiety effect. So yeah, interactions could happen among amino acids, which is a topic that may be relevant to investigate in the future research, for sure.
Another question, Max, for you. It says, look, you're sort of indicating that the impact on meal pattern, feed intake, are related to the gut peptides, as you mentioned, CCK, GLP-1. But obviously with a gavage like the ones you're giving, you will see also some of these, maybe lysine, leucine, going up in plasma. Would these increased plasma levels of amino acids have a direct impact on the hypothalamus or the brain, and therefore potentially explaining the impact on satiety and satiation?
Yep, good question. Yeah. I will expect that some amino acids will have an impact, a direct impact on the central nervous system. It's something that has been described extensively in rodents. There's some information in pigs as well, describing that some amino acids, particularly branching chain and specifically leucine, can alter through the mTOR signal pathway the activation of neurons within the hypothalamus, leading to the sensations of satiety. So yeah, it could be and this could potentially also explain some of the extended effects of the amino acids shown in the in vivo study, because this both lysine and leucine reduced food intake up to 3 hours post-gavage. And considering that the half-life of gut peptides tends to be relatively short, one could expect that potentially the initial effect of these amino acids on satiety may be through the activation of these gut peptides, but then subsequently there might be activation of other mechanisms, post-absorptive mechanisms, such as the activation of neurons within the hypothalamus, which could extend the satiety effect of these amino acids in pigs. So yeah, so my answer is yes, there could be, or we could expect an effect of some of these amino acids in the central nervous system.
One of the attendants to your chat observed that the impact of phenylalanine on GLP-1 was quite delayed. It showed a pattern of after 30 minutes and increasing over time up to 2 or 3 hours. And then this is quite a unique pattern. Do you have an explanation of how this delayed impact may occur? Is that a systemic impact? What is it?
Well, we're not certain. I'm not certain, to be honest, but I can speculate that the mechanism behind the effect of phenylalanine on GLP-1 is different to that of glucose. So in the case of phenylalanine, considering that the increase of GLP-1 happened after the initial half an hour post-gavage, this could be related to the activation of enteroendocrine cells in the distal gastrointestinal tract. I'm talking about ileum and colon, which is something that potentially didn't happen for glucose, because as it was shown, glucose increased GLP-1 levels really quickly within 5 minutes. So potentially there might be a mechanism within the proximal gut that may be inducing the release of GLP-1 in the case of glucose. While in the case of phenylalanine, as I mentioned before, I think this is more related to the direct contact of phenylalanine with enteroendocrine cells in charge of releasing GLP-1. There potentially could be interactions as well, as I mentioned before. There's interaction between gut hormones, and we didn't test or evaluate the effect of amino acids on all of them. So potentially, phenylalanine could be stimulating the release of other gut peptides that could have a positive effect on GLP-1 as well. That's something that cannot be discarded either.
Thank you. Pat Blackwell is asking, I like that question, and I think it's a very, very intriguing one. He's mentioning about this differential impact of responses in the duodenum as opposed to the ileum, based on your ex vivo studies. But also in the gavage, we see responses in the ileum one, and I'm adding that to the question of Pat, because most of the free amino acids you expect will be absorbed before getting to the ileum, right? So Pat's asking, what is the typical transit time between these sites in the live pig, if you know?
Well, I can give approximations. So it has been described that amino acids or nutrients reach the duodenum within a couple of minutes after ingestion. Of course, many of the content may remain in the stomach for many hours for it to be digested. It has been described even sometimes up to 8 or even more hours in the stomach. Some of the content may remain there. But some of the content may quickly pass to the duodenum within a few minutes. So that's within 5 to 2 minutes. Some of the content may be already interacting with enteroendocrine cells in the duodenum. But then the transit time between duodenum all the way to ileum may vary a lot depending on the content of nutrients in the diet. If we're talking about a diet with containing high levels of fiber, that might be completely different compared to other diets that may contain a lower concentration. But I would expect that, considering that the increased levels that we observe for phenylalanine around the 30-minute mark, that I could be potentially, this is to a certain extent speculation, that probably it takes around between 20 to 30 minutes for amino acids to reach the distal ileum. In this case, the amino acids were administered alone, so they were not contained within a diet. So that could potentially also have made it so that the transit is faster as well.
But you would expect the amino acids would be absorbed before reaching the ileum, wouldn't you?
Well, I mean, the concentrations we administered were slightly above what one can find in normal commercial diets. So potentially a part of them could have reached. But as you mentioned, it could be also that there might be an indirect activation of distal mechanisms by stimulation of potentially gut hormones in the proximal gut, or even the vagus nerve. It has been described, at least in rodents, that the proximal activation of the vagus nerve can lead to the secretion of GLP-1 in ileum and colon. So that could also be happening in pig, but then again, it's a little bit of speculation.
Because this question links with another question we've received saying, would you expect protein-bound amino acids to have a similar impact or very different than free amino acids?
Well, I mean, there's quite recent information describing that besides free amino acids, their D and 3 peptides have also been shown to be significant.
I can barely hear you. Sorry, Max. I don't hear you very well.
Sorry. So what about now? Can you hear me now? Yeah. So yeah, I was saying that, yeah, it has been described quite recently that besides free amino acids, some D and 3 peptides have also been shown to be stimulants of gut peptides such as CCK, GLP-1, and ghrelin. So yeah, some amino acids in the form of peptides could potentially also be inducing the release of these hormones. Now, the same has not been described for larger size peptides. So I'm talking about oligopeptides, polypeptides, but for sure it has been described for smaller D and 3 peptides. So yeah, there could be potentially some interesting interactions that could be happening there.
All right, look, let's take these last 2 questions and we'll finish after those 2.
Okay.
One says, refers to the anesthesia. Could the use of anesthesia have influenced the satiety signaling? It was not the general anesthesia, was it?
No.
But so nevertheless, even though it was not a general anesthesia, as the question has been framed, it was not a general anesthesia, but still, would anesthesia influence that satiety signaling independently of the effect of the gavage treatment? That's a good question.
Yeah, that's a really good question. So, yeah, first of all, we only lightly anesthetized pigs. So once they started to show the first signs of sedation, we then cut the anesthesia completely. We administered the gavage, and then we just, returned the pigs to the respective pens. But in regards to the anesthesia, yeah, we collected samples not only just before the gavage, but also before the anesthesia. And when looking at the concentration of CCK and GLP-1, there was no big difference between the concentrations of the blood collected before the anesthesia or after the anesthesia, but before the gavats. So we pretty much concluded that potential, if there is an effect, potentially is quite minor. So we went ahead and so I don't think it was a big, the anesthesia had a big impact on our results.
Okay, so I'll leave you off the hook after this last question, Max, which comes from someone well known to us. Sharam asks, according to you, what would be the next question in terms of research? An interesting question for research, maybe, and he proposes related to non-essential amino acids.
Yeah, that's an interesting question, because as I showed, there's some amino acids related to gut health, such as glutamine, asparagine, glutamic acid, which were shown to induce the release of these peptides. And now it's important to mention that CCK and GLP-1 are not only satiety peptides; they play other roles in the digestive, in the regulation of the many digestive mechanisms. So they also reduce intestinal transit, stimulate the gallbladder contraction. So they aid on the digestion and absorption of nutrients. So there potentially could be a benefit of, testing these amino acids and their impact on these hormones and how they could potentially be used to improve gut health in pigs. And as for the next step for investigation, I think, yeah, there is still a lot that we don't know about the effects of amino acids on gut peptide and appetite. I think it would be interesting to evaluate with more detail the mechanisms behind these processes, not only the amino acid sensors that may be involved in the transmembrane of enteroendocrine cells, but also as well some of the pathway within or inside these cells as well, which has not been properly studied in pigs yet.
Alrighty, well done, Max. Thank you so much. That's been a very good presentation, excellent presentation, and a good set of questions. If anyone has any additional question, of course, just contact Max or myself after this. So without further delay, I would like to announce the next week's QAAFI Science seminar. That will be on Tuesday, the 27th of July, and it's titled Fractals, Plants, Insects, and Orchards, Reflections on Life with Health Systems, and will be presented by Jim Hannan from QAAFI's Center for Horticultural Science. If you need additional information, please do visit the QAAFI Science Seminars webpage. And with that, I would like to just encourage you to participate in the poll at the end. And if you haven't already signed up for the seminar invitation list, please do that at the quaafi.uq.edu.au Science Seminars. And with that, I'll thank you all for your attendance. Thank you, Max, for an excellent presentation, and bye-bye to everyone. Bye-bye.
Thank you very much. Bye-bye.